In this video, we will discuss about the Sialoglycan and Siglec interaction that cause the immune suppressions. We have understood one of this example in detail when we are discussing about Siglec-10 interactions with its corresponding ligand on the pancreatic cancer cells. So, we will discuss these things in general, like how this Sialoglycan and Siglec interactions in the innate immune cells and also in the adaptive immune cells cause immune suppressions. We will start with this adaptive immune cells, which is this T cells. The T cells, activated T cells usually express the Siglec-9 receptors, which can be interacted with with the Sialoglycans that are expressed on the tumor cells, and once they are interacted to each other, it will cause the inhibitions of the T cells activations. Thereby, T cells will not get activated further, and this will this will suppress the immune response by the T cells against the immune cells, by the T cells against the tumor cells, and that now in terms of this innate immune cells, this NK cells can express the Siglec receptors Siglec-7 and Siglec-9, and this Siglec-7 and Siglec-9 can also be interacted with its respective Sialoglycans on the tumor cells, thereby prevent the NK cells killing on the tumor cells. And in terms of this myeloid derived suppressor cells that can trigger phagocytosis, and those phagocytosis can be reduced by interacting with this Siglec-10, which are usually expressed on the myeloid derived suppressor cells with the tumor cells. So, and we have discussed one of these examples where we have talked about where the researchers has find out the respective ligands that can interact, major respective ligands that can interact with the Siglec-10 in the pancreatic cancer cells, and they find out that they are the subunit of integrins, and by preventing the interactions between Siglec-10 and integrins on the tumor cells, the researchers able to show that it will enhance the phagocytosis of the macrophage against the tumor cells. At the same time, it will also activate, or it will prevent the inhibitions of the T cells that we have discussed with the enhanced T cells differentiation. And at the same time, we have also seen that the granzyme that are released by the activated T cells has also been increased. So these interactions will not only cause one kind of suppression, but it might have a cascade effect. So by preventing those interactions, people can increase the immune response against the tumor cells. At the same time, it will also activate the other immune cells to act against the tumor cells. So, and in terms of these NK cells, the Siglec-7 and Siglec-9, two Siglecs are expressed. So that we have just that I have already said you that these two Siglecs are expressed. So they are more prone towards immune suppressions compared to the other immune cells which has like single receptors. For example, these single receptors can interact with this Sialoglycans that are expressed on the tumor cells. For example, the T cells has Siglec-9, and these myeloid derived suppressor cells or MDSCs have this Siglec-10. So, and it is not necessary that only this this carbohydrates based interaction occurs. The non-carbohydrates based interactions are also occurred, and that are widely known, such as these PD1 and PDL1 interactions, and this CTLA4 also interacted with this respective ligand on the tumor cells. So those are the non-carbohydrate based interactions. So those also cause the immune suppressions. So, but those things are those non-carbohydrate based these immunosuppressions receptors are already well known and well researched, and people have find out a way to prevent those interactions. But this carbohydrates based immunosuppressions receptors are still in the phase where people discover some new receptor ligand interactions, and by suppressing those interactions they are able to see enhanced anti-tumor efficacies. So far it has given a positive results. However, right now, because of the limitations in detecting what are the different kind of glycans or glycoproteins are present in the samples, it is difficult to reach to a point where people can find out what are the different sialoglycans and Siglec interactions occurs. What are the different sialoglycan Siglec interactions occurs within immune cells and the tumor cells. So if this information has been known, then researchers can increase the anti-tumor efficacy significantly. So right now this is in the process of research. Hopefully in future we will have the more knowledge about those interactions that can be used as a therapeutic target to enhance anti-tumor efficacies. So that's all for this video. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. Thank you for your time.